Hi and welcome back to another video, and as promised in my previous video, I said I was going to explain more on C3 and C4 plants and how they carry out photosynthesis very efficiently. Now, plants have evolved different strategies to carry out efficient photosynthesis and survive in their environments. Now, while most plants are C3 plants, which thrive in cold and moist environments, plants in hot and dry environments have developed special adaptations, and these enable them to thrive in hot and dry environments. And these undergo the C4 pathway. So in this video, we shall compare C3 and C4 plants, looking at their structures, adaptations. Now, if you have watched my video on photosynthesis, you will understand this one very smoothly. Now, with all that said, let's get into it. Now, C3 plants include soybean, or in Africa here we call it soya bean, tomatoes, wheat, and these are basically all legumes and cereals. And these thrive in cool and moist environments. Now, as most of us know, C3 plants fix carbon dioxide directly into the Calvin cycle, forming glycerate phosphate. However, these suffer two major disadvantages in hot and dry environments. Firstly, C3 plants must open their stomata to obtain carbon dioxide for photosynthesis take place. However, when the stomata are open in hot and dry environments, they not only allow carbon dioxide to enter the plant, but also transpiration occurs and therefore water is lost. So C3 plants have low rates of photosynthesis in hot and dry areas because they have to also close their stomata to avoid this excessive loss of water, and this reduces the amount of carbon dioxide diffusing into the plant. And the second disadvantage that C3 plants face, which is also the main disadvantage, is photorespiration. Now, C3 plants have ribulose phosphate as their carbon dioxide acceptor, and this fixes carbon dioxide and converts it to GP, or glycerate phosphate, in the presence of rubisco, and this rubisco catalyzes this reaction. Now, rubisco is an enzyme and it has a higher affinity for oxygen compared to carbon dioxide, leading to photorespiration, and this reaction is actually wasteful. Now, before that I talk about this, you should know that photorespiration is the process in plants in which rubisco binds to oxygen instead of carbon dioxide during the Calvin cycle. And this is wasteful because when Ruby binds to oxygen, it catalyzes a wasteful process, forming what is known as glycolate or glycolic acid, and this is a 2-carbon compound which is unusable by the plant. Now, this glycolate, since it's of no use to the plant, is therefore recycled to glycerate phosphate through the chloroplast, peroxisome, and mitochondria in what is known as the glycolate cycle, or the C2 cycle. Now, what happens here is the glycolate leaves the chloroplast and diffuses into the peroxisome. Now, I know this organelle is usually ignored, but it's actually very important. So once the glycolate is in the peroxisome, it is oxidized to glyoxylate, and this glyoxylate is then converted to glycine. Now, this glycine then diffuses out of the peroxisome and enters into the mitochondria. Now, once inside the mitochondria, two molecules of glycine combine to form serine, and in the process, ammonia and carbon dioxide are released. Now, this serine diffuses back to the peroxisome, where it is converted to glycerate. This glycerate finally diffuses back to the chloroplast, and ATP is broken down. Now, the phosphate from ATP is added to this glycerate to form glycerate phosphate, and the Calvin cycle occurs normally. Now, recycling glycerate to recovers glycerate phosphate, and the carbon dioxide that is released is refixed by Rupi. But this uses out of energy, so you realize that photorespiration decreases the rate of photosynthesis and reduces productivity in C3 plants. So with that said, you realize that in hot and dry environments, carbon dioxide levels are very low, so C3 plants don't usually grow well, and I hope that is clear. That said, we can now move on to C4 plants, which include sugarcane or maize, which is also called corn in some regions. Now, C4 plants survive in hot and dry environments because they have evolved some adaptations which reduce water loss and also reduce the rate of photorespiration. Now, the first adaptation is that C4 plants have what is known as the Kranz anatomy, and this basically means that C4 plants have chloroplasts, have chloroplast-rich cells which form a ring-like pattern around the fascicular bundles. Now, the cells in the outer ring are called mesophyll cells, and the cells in the inner ring are called bundle-thick cells. So, what you need to know from here is that structure determines photosynthetic efficiency and productivity. Okay, structure determines photosynthetic efficiency and productivity. So the difference in structure in C4 plants is that gives them the edge over C3 plants. Now this Crohn's anatomy in C4 plants gives them the edge over C3 plants in hot and dry environments. So this arrangement separates the initial carbon fixation by PEP in the mesophyll cells and the Calvin cycle in the bundle cells. Now in the mesophyll cells, C4 plants use PEP to fix carbon dioxide, forming oxaloacetate, and this reaction is catalyzed by PEP carboxylase, which has a very high affinity for carbon dioxide and no affinity for oxygen, so it does not combine with oxygen under any circumstance. Now, because PEP carboxylase has a high affinity for carbon dioxide, this means that when oxaloacetate is converted to malate and this malate transferred to the bundle cells, you realize that the malate loses carbon dioxide to form pyruvate. But the point here is that the PEP carboxylase has a high affinity for carbon dioxide, and this means that there will be a higher concentration of carbon dioxide released in the bundle cells, and this carbon dioxide is fixed again by RuP in the presence of Rubisco in the bundle cells, and the Calvin cycle proceeds. Now, the Rubisco in bundle cells works at an advantage compared to the same enzyme in C3 plants, and this is because of two main reasons. Firstly, Rubisco is not competitively inhibited by oxygen, so photorespiration does not occur, and even if it occurs, it's almost negligible. And the second reason is that Rubisco works more efficiently at higher concentrations of carbon dioxide. So here what you need to know is that Rubisco is an enzyme, and as we know that enzymes increase the rate of reaction when the substrate concentration is high. So Rubisco works more efficiently at higher carbon dioxide concentrations. And with that said, our second adaptation is actually very clear, and that is that C4 plants have PEP carboxylase in the mesophyll cells, which has a high affinity for carbon dioxide and no affinity for oxygen. So this means that little or no photorespiration occurs. Now the reason why I've put little is that we cannot ignore the fact that there is oxygen in our atmosphere, and therefore some of it diffuses through the stomata into the leaf. So I'm sure it somehow diffuses all the way to the medullary cells, and a small fraction of this oxygen binds to Rubisco. But due to the high concentration of carbon dioxide, the oxygen is outcompeted by carbon dioxide for the active sites in Rubisco. So some photorespiration still occurs, but it will almost be negligible and does not affect photosynthesis. Okay, now another adaptation is that C4 plants can keep their stomata more partially closed compared to C3 plants and still obtain enough carbon dioxide. So this enables C4 plants to use water efficiently since less water is lost, and this helps them to survive in hot and dry environments. So these adaptations enable C4 plants to outcompete C3 plants in hot and dry environments, but not in cool and moist environments. Now in cool environments, C3 plants have the advantage over C4 plants, and this is because in cool environments, Rubisco naturally has a higher affinity for carbon dioxide than oxygen. So there will be a lower rate of photorespiration, and the double fixation of carbon dioxide in C4 plants by PEP carboxylase and Rubisco basically uses extra energy, and C3 plants can grow faster because they spend less energy in carbon fixation. So C4 plants only age C3 plants due to their ability to reduce the rate of photorespiration. Also, in cool environments, C3 plants do not lose a lot of water by transpiration, since despite the stomata opening for carbon dioxide to diffuse into the leaves, the cool and moist environment reduces the rate of transpiration, so they can conserve more water, and this helps them to carry out photosynthesis more efficiently in cold and moist environments. So with all this, you realize that in hot environments, C4 plants win because they reduce photorespiration, and in cool environments, C3 plants win because they fix carbon dioxide efficiently without using extra energy that is needed in C4 plants. So please do, I request you guys to do research on CAM plants, and this is not very different from C4 plants. And with all that said, I believe this has been of some use to you guys. So please subscribe, like the video, and please again subscribe, and I'll see you in my next video. Thank you for watching.